One final way that's new and is promising is called the RNA vaccine. With RNA and DNA, instead of putting that shape in, you put instructions in the code to make that shape.
So this is a precision tool that now allows us to take this protein RNA complex and introduce it into cells or tissues to correct mutations at sites where we know there's a deleterious change in the genetic code. So I wanted to show you an example of how this can be utilized. This protein complex can actually be injected directly into fertilized eggs of a mouse. And in the experiment you'll see, we're targeting a gene that is responsible for the black coat color in mice. And so normally these mice have beautiful glossy black coats, and once we make this targeted change, we then implant these edited eggs back into a female mouse, and when she gives birth to pups, you'll see that the pups are now mostly white. And the remarkable thing about this experiment is that when these mice grow up, you can test them and show that every cell in the body has this single genetic change that gives rise to the white coat color, but otherwise they're absolutely normal. So they are normal mice, no mutations elsewhere in the genome. This is a type of experiment that used to take at least a year to create a mouse like this, and now it can be done in a few weeks and by people that don't have to have special expertise. So this is a technology it's been very exciting over the last 2.5 years to see this technology taking off.
I think we will see a lot of advancements with mRNA. And the good thing, after the success of COVID, it is that right now we have a substantial wave of companies, biotechs and big pharmas that are working on mRNA. A lot. And they are working on multiple applications of mRNA. So I think the first things that I'm hopeful to see it is other vaccines other than COVID. A flu vaccine with mRNA, a single vaccine with mRNA, other vaccines with mRNA.
We think within the not too distant future it will be possible to actually use this technology to make changes in humans.
One of the features of this fourth industrial revolution is that it doesn't change what we are doing, but it changes us.
The difference of this fourth industrial revolution is it doesn't change what you are doing, it changes you. If you take genetic editing, right. Just as an example, it's you who are changed. And of course, this has a big impact on your identity.
The first applications of the CRISPR technology are going to happen in the blood, where it's relatively easier to deliver this tool into cells compared to solid tissues. Right now, a lot of the work that's going on applies to animal models of human disease, such as mice. The technology is being used to make very precise changes that allow us to study the way that these changes in the cell's DNA affect either a tissue or, in this case, an entire organism. Now, in this example, the CRISPR technology was used to disrupt a gene by making a tiny change in the DNA in a gene that is responsible for the black coat color of these mice. Imagine that these white mice differ from their pigmented litter mates by just a tiny change at one gene in the entire genome. And they're otherwise completely normal. And when we sequence the DNA from these animals, we find that the change in the DNA has occurred at exactly the place where we induced it using the CRISPR technology. Additional experiments are going on in other animals that are useful for creating models for human disease, such as monkeys. And here we find that we can use these systems to test the application of this technology in particular tissues, for example, figuring out how to deliver the CRISPR tool into cells.
Other vaccines other than COVID, a flu vaccine with mRNA, a single vaccine with mRNA, other vaccines with mRNA.
Using CRISPR to help cure disease is not very controversial, but using CRISPR, you could actually change the DNA that would determine your baby's eye color or skin tone.
This is no longer science fiction. Genome-engineered animals and plants are happening right now. And this puts in front of all of us a huge responsibility to consider carefully both the unintended consequences as well as the intended impacts of a scientific breakthrough. Thank you.
Jennifer Doudna, the Royal Swedish Academy of Sciences, has decided to award you the 2020 Nobel Prize in Chemistry to you for the development of a method for genome editing. It is a great honor to convey to you my warmest congratulations and ask you to receive the Nobel Prize.
Walter Isaacson's new book, The Codebreaker, tells an amazing story. It's about how Jennifer Doudna and other scientists discovered a new way to edit DNA. Editing DNA with precision has been a holy grail for scientists for decades. The DNA, of course, encodes all of the biological functions. And so some of the mistakes in DNA cause disease, genetic disease. If we could go in and fix those mistakes, we could save many, many lives and get rid of these diseases.
CRISPR technology was used to disrupt a gene by making a tiny change in the DNA in a gene that is responsible for the black coat color.
The experiment you'll see, we're targeting a gene that is responsible for the black coat color.
It changes you if you take genetic editing. Right. Just as an example, it's you who are changed.
The first applications of the CRISPR technology are going to happen in the blood, where it's relatively easier to deliver this tool into cells compared to solid tissues.